You were here yesterday, so you know where I'm from, but I can give, because I've got longer to talk to, and give you a better introduction. I'm not going to talk much about well, I'm not going to talk anything about FBA, and I'm not really going to talk about anything about microbial communities again, unless you count the mitochondria and the nucleus as a community, which I guess it is. My major focus in the research is looking at how energy and metabolism feeds back on transcription, and I'm going to talk about a little pathway or system that we found in yeast that regulates how transcription is reset during the yeast life cycle. I'm from Keio University in Japan, and I work in an institute in, so this is Keio University, oh, wrong one. So this is Keio University. It was founded by this chap here, Fukuzawa, this is one of the oldest buildings there in the Mita campus. We're based mostly in Tokyo, and this guy's one of the most popular guys in Japan because he's on the 10,000 yen note. Everybody loves him. Nobody doesn't like him. But he was a great modernizer, and during the Meiji Restoration, he basically introduced modern university systems into Japan. So this was the first modern university in East Asia. Our institute was set up by this fella. This is Masaru Tomita, whose idea was to quantify. He's a computational biologist. He developed natural algorithms for inputting languages. But his idea was to set up an institute to quantify and model everything based in the countryside in Japan, which is here. This is the computational arm. They get a nice fashionable building in the centre of town. We're working in the rice fields and the metabolome factories in the rice fields. So this is the metabolome factory, which is about 4 kilometres from here, and this has over 100 mass specs set up in various configurations to monitor various things, which is a great resource for what I like to do, which is quantify stuff. So basically, I'm in QBio. The institute itself is in a really nice area of Japan. We've got the beach really close. This is part of the same town. This is kind of the view from my house during springtime. And this is like a human incubator. And the best thing about the town is on the 25th of May, which coincides with a very famous person's birthday, they have a big, huge sake festival. And the girl in the background there is my daughter dancing for the festival. So I really quite enjoy a relaxing, quantifiable time there. So the technology we use mostly for quantification of metabolites is capillary electrophoresis, mass spectrometry, where basically we can have a charge here on the CE side, positive and negative, and then this separates metabolites, in this case cations, out by their charge and their molecular volume. It also rips off adducts, so we get very little in the way of adduct formation. So we don't have very complicated spectra. This is currently what we're using to do anions, which is kind of a reverse system of this, where we take the silica coil and put a patented layer on, which assists in swapping the charge around and then determining anions. So to give you an idea of what we can measure, we've got all the stuff which is the good standards, and we can measure most of the anions, including ATP, GTP, nucleotides, and the cations, which are amino acids and stuff like that. And we can quantify them because we've got standards.
So the advantage of capillary electrophoresis is it's small sample size. What's the advantage of?
The advantage, there's 3 major advantages of capillary electrophoresis and a couple of disadvantages. The main advantage is that it rips adducts off, so you don't have adducts forming with the analyte. So the analyte in normal LCMS is very difficult because your metabolite of interest or your compound of interest forms ammonia or formate or whatever's in the buffer media, and it can form multiple adducts of these, and even chains, and it makes it really difficult to say that's what I want, that's what I have. The other advantage is speed and sample volume. Sample volume's 5 microliters, maybe less in this. The other advantage is it's not per se separating by size, which most LC techniques do. It's separating by charge and molecular volume, which gives a greater range. So, for example, ADP here is much larger than lactate, and it comes at about the same time. It's all compressed into one size. We don't need to wait for ages for ATP to come out. Okay? Okay, so.
The disadvantage, what's the disadvantage?
The disadvantages, one of the main disadvantages are that you need to tune it very well. Tune the parameter, and the column, especially on an anions, the column degrades, and when the column degrades, you start to get period drift. So it's difficult to align chromatograms, which is a pain in the earth. Okay, so what I do there is I run continuous culture. With the aim of elucidating yeast physiology and how yeast work. So, this is the software that I run. This is an old version actually. Kalesh might recognize this. Basically, we've got. You can write a driver in HTML and control any instrument that has a communication. And then as long as the parameters are in the XML file, if the XML file is compatible with this, then you can control any instrument that you put on. So, for example, I've got the codes for the Agilent bioanalyzer. I can use this for the Agilent bioanalyzer. And I can hack instruments as well. But anyway, so basically continuous culture, for those who are not au fait with it, is you pump through media. This is a diagrammatic representation of it all. You can pump through media. You control this reactor very precisely, and then there's an outflow of the media, and traditionally it's always thought that this goes into a steady state. However, when bacteria, yeast, cell lines go into a certain cell number, they start communicating with each other, and then they can produce very stable but highly dynamic systems. So a stable dynamic system. There's also other neat things we can do with this. There's a scripting language down here where we can program things to do with the fermenter, like gradients, pulses, inject noise, make whatever you want. There's a simple math parser that you can do simple mathematics of adding instruments together to make a scenario. So that's the culture. Highlights include this here is a water bath which controls the temperature here, but by monitoring this temperature and using Fourier's equation we can calculate the heat produced. So we've got a calorimeter here. The sodium hydroxide addition, we can calculate the hydrogen efflux from the system, so we can get overall hydrogen ion fluxes from the system, and we can find out when people are near the fermenter as well. Now, this is the idea of the stable dynamics that I have been studying for a while now. Basically, you've got a very stable oscillation which can last for months. These are just 2 short runs of about a week each in 10 days. And basically, you've got a high amplitude dissolved oxygen, residual dissolved oxygen oscillation, which switches between a highly oxidative state to a highly reductive state. So this is zooming in on one of these cycles. And this is highly repetitive, and it means you can stitch together data taken over many years, because you've got this relationship between dissolved oxygen and your measurement. So we can stitch together data that was taken from 15 years ago and add it to data now, and it's still relevant because it's the same system we're studying.
Once again, what's the time of an oscillation?
Okay, wait a minute. So
the doubling time in that setting?
The cell division time in this system is 8.1 hours, but with 8.1 hours, you can the dilution rate set to 0.085, which gives you a cell doubling time of 8.1 hours, and you can get a Thaler period of anywhere. I've got between 0.5 and 8 hours for Saccharomyces cerevisiae, just by varying strain. You've got mutants that you can use, or other model Saccharomyces cerevisiae strains, or you change the conditions of the media in. So it's environmental and genetic. You can change the period considerably.
Sorry, if I keep asking. What's the range of dilution rates?
This is set at the same dilution. This is usually set at the same dilution rate. I have done dilution rate experiments, and it's not as linear as you might expect.
Yeah, I mean, this seems to be, fairly low resource kind of environment, right?
Yeah, high cell number, low resource.
Yeah. So what if you push it towards higher dilution rates, more resources?
You change the period, but not in a linear way.
Okay.
It switches basically. You've got modes of stable periodicity. Okay. But I'm not showing that data here, so sorry.
Okay, yeah.
I can show it to you at some point in the future.
But what's the thing that oscillates, the cell number, or?
No, dissolved oxygen. Well, dissolved oxygen. So we call it the respiratory oscillation, because it encompasses much more than that, because I'm about to show you what it encompasses in the analysis that we can do on this. So, as you said, it's a low nutrient environment, so there's actually nothing in the way of exometabolome. There's extracellular proteins, but nothing really oscillatory, so it's not like corm sensing, like in where a bacteria releases a protein. It's sensed and then they do some activity together. What does oscillate quite well is the gases in the system. Let's call it the gasome, why not? So you have acetaldehyde for sure. I can show you plots of this to anyone who's interested. We did phase response curves for all of these. And possibly carbon monoxide, but carbon monoxide is a bit funky because it does the opposite to the respiratory chain that you might expect. It appears to be acting as a substrate. So respiration is actually enhanced when you add carbon monoxide, and we're not too sure why. But carbon monoxide could be important. Amplitudes modulated by other gases, hydrogen sulfide, oxygen, carbon dioxide, and ethanol, which is not really gaseous at 30, but it's got a high vapor pressure, so it's kind of semi-gaseous. And you can modulate the frequency by changing the ethanol and glucose flux. You can change it by adding an endogenous ROS producing chemical. And you can also, wrong one. Back. Okay. And you can also, interestingly, change it with changing one vitamin, pyridoxal 5-phosphate. You can change it's a very dynamic thing, and I've not got time to go through it because you get lots of bifurcations and chaos in that experiment. But these kind of appear to modulate the period.
You mean that when you add acetaldehyde, you can tune the phase of
Yeah, you can shift the phase forward or backwards, like a traditional phase reset clock. So acetaldehyde is very quite important. What we can measure online, this is just an example of what we can measure online. It's all related to phase angle now. I've taken the time out of it because if it's so reproducible, we can compare things just with phase angles rather than doing time. I'm not used to this. High tech. So basically, we can. This is a dissolved oxygen of 20 cycles, and then there's a linear fit, a spline fit on this. And basically, the QH is monitored by the sodium hydroxide addition to the system to control the pH. And then from that, we can calculate how much hydrogen ions were released by the bacteria. Q heat's calculated with Fourier's equation and the two temperatures. So we can see that when respiration activity is high, you have a lot of hydrogen efflux and you have also a lot of heat production, which makes sense. This is oxygen production rates, so it's the opposite. So this is where oxygen's high. Oxygen uptake's high. It's flipped because we're doing FBA, and it makes it easy just to make it negative because it's actually consumed. And this is CO2 because we can monitor this in the off gas. Hydrogen sulfide, we've also got a hydrogen sulfide electrode, and we can also measure NADH, NADPH using fluorimetry. So this is a kind of detail that we can actually specify what cell state, what state that culture's in at that particular moment in time.
This sulfide, where does it come from? How much do you find?
We find that the cells produce 0.020 millimoles per hour per gram of cell. These are all in millimoles per gram per hour of cell, except heat, which is in watts.
So where's that linked to? Where's that coming from actually?
Well, the hydrogen sulfide, sulfate uptake.
Okay.
So you've got sulfate uptake, and it's an intermediate of sulfate uptake, hydrogen sulfide. And then if you've not got enough homoserine in the system, or there's a block, or it's inhibited somehow, then hydrogen sulfide will be released. So I've got another talk about that, but I'll not talk about it.
Isn't it toxic to the cell?
Pardon?
Hydrogen sulfide, is that not toxic?
And not at these concentrations. The phase response curve is interesting. It only really affects the concentrations of hydrogen sulfide. Only really affect it when it's high. When you've got a high concentration of hydrogen sulfide, you add more hydrogen sulfide, and the system increases in amplitude. So it's not really a synchronization agent. A synchronization agent would mean that you'd be able to shift this. If you injected hydrogen sulfide here, you'd be able to shift the entire oscillation forward or backwards, depending on when you inject. I should have put that graph up. I could.
Is that in serious levels, or like also just in very low levels?
The acetaldehyde is in about. The acetaldehyde is about 10 times higher.
Okay, but it's not a significant proportion of the flux that goes out, or is it?
Not really. It's more of a signaling. But ethanol is an important part of the flux. So the flux through. So ethanol's always produced in the system, unless you're growing it on ethanol, because you can get the same system growing on ethanol and oscillating. But ethanol's always produced on glucose. Okay.
But then it's also taken up again.
Yeah. So you've got I should have put that graph up
So
as
it's
well.
like acetaldehyde, like synchronized with ethanol?
No, it's out of phase. It's almost 180 degrees out of phase.
Okay, so basically when you're taking it up, you produce acetaldehyde, or?
Yeah, because I think that flux is, yeah, it makes sense. Yeah. Okay, so this is just background, so the cytometry I went through yesterday, but I'll go through it again. Will I? No, I won't. I'll maybe come back to this one. But the cell cycle is synchronized. The S phase is synchronized during the oscillation. So this is S phase here. This is a traditional histogram from flow cytometry. So you've got G2 here, S phase, and G1. And the cell progresses. You can just see it here, this light blue line. But then when you do a Fourier cleanup on this system, you can see that S phase occurs almost throughout the entire cycle, but it stops here. This is the point that I want to focus for the rest of the talk, or for much of the rest of the talk, this point here. We're at the transition between late S phase and early S phase, where the system flips. Okay, this point here.
Mitochondria, do they divide at a certain
Later. That's my last slides. I can't go to. Do you want to reorder my talk? Okay. So
You have a couple. So just to understand, it's okay to interrupt you, I guess, right?
Yeah.
You have a couple of oscillations before a cell divides, right?
You've got about the cell division time is 8.1 hours. This oscillation is 40 minutes. So you've got about 11.7 oscillations before. So let's call it 12.
Okay. But so both your life cycle or division is synchronized, and your oxidation cycle or whatever it is.
Yeah, so this is only you can see that this is a very low percentage of the cells.
Yeah.
So you've got about 8 to 10% of the cells going through S phase at any particular moment in time, and this is linear. This confirms that S phase is a linear thing. So once S phase starts, it doesn't stop.
Okay.
And it progresses at a linear march through the DNA. And there are early firing and late firing origins of replication, the ORCs. Okay, so if I've got time, I can tie that in with some DNA data, but I don't think I'll have time. Okay, so, but interestingly in the system, the dry cell weight never really changes, the cell number doesn't really change. This pink line, there is if you've got the right oscillation goggles on, you can see that the distribution of this changes. So the bottom line is the lowest distribution, the top line is the highest distribution, and the red line is the median. But if you've got the right goggles on, you can see that this kind of oscillates, but I wouldn't bet money on it. Now, one of the first results we got about 8 years ago now, 8 years, is that we did microarrays on these, and the entire transcriptome was also flipping around. So this is just a simple analysis about how many cycles are involved. We basically got a state where these genes are switched, these genes are activated, and then it flips to a state where these genes are activated, and it's throughout the entire cycle. This is a phenelazine injection, which is an antidepressant, and what that does is damp the oscillation down and double the period. So this is a double dynamic system. So basically the entire transcriptome oscillates with quite a high significance. So you can see during the oxidative phase here we've got a lower number of genes generally. The proportion of this doesn't change. And then these guys here are the noisy genes. So blue here means noisy. This means oscillating with significance. Okay? And then blue means noisy. So there's only about 10% of the genome which isn't doing anything, which was quite a big thing. So we thought, wow, what's all this about? Luckily, or unluckily at the same time, another group released a transcriptome data on a 300-minute period, 5-hour period. So it gave us the opportunity to do some sort of cross-analysis between the two data sets. So basically this is just a summary of the cross-clustering we did. We used, because the data kind of looks like flow cytometry, we used a flow cytometry cross-clustering, a flow cytometry algorithm called FlowClust, and then that's a Bayesian inference measure, and you can basically tell how many clusters are optimal from the cluster distance and the Bayesian inference coefficient. So the optimal number of clusters between the two data sets was actually much higher than we expected, about 11. But the top oscillators were all in this capital letter stuff. The strongest signal oscillators were all in this capital letter stuff. So the clusters A, B, B, BC, C, D, BD, and D were all in the top. And these are the time series actually plotted, so you can see that they're very oscillatory. Now these came out in phase space. So this is the phase angle for the 40-minute oscillator, and this is the phase angle for the 500-minute oscillator. We can see that the cluster A is concentrated here, and then it goes through a progression. Now this is multidimensional, so there is some overlap between the two. Multidimensional, I mean, we took the significant Fourier powers, the significant real and imaginary components from the Fourier, and then clustered those to scale down the problem, and then that was used to construct the clusters. And that means it's a circular clustering as well, because if you start clustering like a normal time series, these guys and these guys are actually in the same cluster, and they wouldn't be in a so it needs to be a circular clustering, just like clustering on a sphere. So to simplify this out, I've invented this little scheme, which is just today. We've got this basic transition from A, B, B, C, D. These are the important ones. These are differential clusters between the two data sets and comprise of about 200 genes. So there's only 200 genes different between the two. So the first thing that everybody does with this stuff is just throw in an ontology search. And the ontology was amazing. Basically, we could get like all the Pol2 was being expressed in cluster A. Ribosomal biogenesis is like 171 out of 199 transcripts. So this kind of became like the ribo cluster. So this is ribosome formation. So A, B, and B were more glycolysis, gluconeogenesis, and amino acid biosynthesis in transcription. Then that switched to a mitochondrial clusters here. So this is quite poor, but basically this is more metabolic. And then the mitochondria ribosomes are being produced, and so on and so on and so on. So we've got and this is the stress response here. So the fungal stress response and catabolism. And basically, once we put the thing in the context of the clusters, a very clear program started emerging. So this is the same thing, but put into the same little "How We Won the War" cartoon. So basically, we've got cytosolic ribosomes, anabolic genes, and anabolic genes being produced. These anabolic genes are more to do with stuff coming off of gluconeogenesis and glycolysis, like serine and stuff like that, and precursors to amino acids. These ones are, and also sulfur amino acids and branched chain. These are other amino acids. Then it goes into mitochondrial ribosome production and mitochondrial genes, genes that are in the mitochondria, not genes that are produced in the mitochondria. So these are like catabolic genes, like your entire TCA cycle and all sorts of stuff. And then this is a differential cluster here, which is another catabolic cluster. So these are kind of mostly located in the symbiont, the mitochondria, and these guys are all located in the eukaryote, the host. So you've got this separation of what the cytoplasm's doing and what the mitochondria are doing. It then switches to what programs are involved. This is just the general growth and stress response, which time points are correlate with growth and stress genes, which have been figured out in Saccharomyces cerevisiae. So we can see that we're going from a growth program to pretty much well a stress program. So we're going from growth here and stress. And the cell cycle is differentially expressed in both systems. So the cell cycle's got much more, anyway, I'll talk about that later. So the p-values are really high. This is just an example of how high, you can't even see it. This is the growth response. So the p-values on the hypergeometric distributions are down to the minus 23. So anything with minus 23, anything in black is highly significant. So you've got, these are really, really enriched in these parts. So this is the growth response. This is environmental stress response. You can see how it all falls out. And then you've got transcription. You can do all this thing, and it's in the paper if anybody's really interested in the numbers. But I thought the summary diagrams might be a bit more informative to get an entire picture of what we're looking at rather than the statistical maps that we did in the paper. So basically, when we're looking at the idea was that it was so general that it had to be something. The real point in the research was that it was so general, it had to be something a bit bigger than a transcription factor coming in. So this is the whole genome flipping. So we had the idea of looking at the bioinformatics of the other data sets to see what chromatin was doing. So this is the architecture of the promoter, where we've got large nucleosome-free regions here in these two components, and a large nucleosome-free region here, and then these are bounded by fuzzy. So anything to do with anabolism and catabolism seems to be much fuzzier. By fuzzy nucleosomes, I mean they're not well defined. By a well-defined nucleosome-free region, you have a very precisely defined area where the nucleosomes can be. So we have a thing there, but this doesn't separate time, because we've got a nucleosome-free region here and a nucleosome-free region here. We've got focusing occurring in these two. Next, we looked at the binding of the chromatin remodelers, and we found that the RSC, which is a remodeling complex, binds pretty much well to all of the clusters, except this one, and this is mostly transcription factor and RSC activity. So you've got the transcription factor comes down, opens it up, and then the RSC holds the gene open for transcription. Except this one, cluster D, was highly enriched in ISWI1. Now this one shuts genes off. So when that goes on the promoter, it shuts genes off. So the action of the RSC complex is to hold promoter structures open. So it bends the DNA one way and holds the promoter structure open, and then that can be transcribed. The ISWI1 twists the DNA the other way and shuts it off. So you've got a differential action in these two. Now both of them are ATP dependent, so we started to think about that. And also we've got, these are the binding motifs which are involved. Basically we're going from the host program to the mitochondrial program. So this is AAT, this is the Ruby box for producing a ribosome. And then it goes to a TATA system, where the TATA box is bound by SAGA. And then it's the same thing for the mitochondrial cohort. And these are controlled by different transcription factors. The CTF are differential transcription factors, and then TF is a big transcription factor network which only really acts at one point in the cycle. So we then thought that this was really interesting, and we did a big metabolomic search for it, and the metabolites showed the same pattern, so the entire network was tuned. However, the metabolism was out of phase with transcription, which makes sense. So you produced the transcript, and then the metabolite appeared 10 to 20 minutes later, once the protein had either been activated or appeared. So this is just all the calibrated standards we have, and their phase relationship with dissolved oxygen. And then this is a few of them that are pulled out just to show you we can quantify things at an attomole per cell basis. So this basically produced the same thing, but kind of so glycolysis appeared kind of here, just before the TCA cycle was activated. And this is where the glycolytic genes start appearing and gluconeogenetic genes start appearing. And then ethanol is produced here, but the ethanol production system appears before. So it's like a chain of events that occur throughout the system. And then amino acids and lipid precursors were later. So this was really interesting. So we've got the phase differences, we can define the phenotype, and we're quite happy with that. But we began to ask ourselves, what's the mechanism in this? What can we do to find out the mechanism? I mean, ATP is a big clue here, because we suspected the ATP remodelers, and that is part of the story, but we found something completely different than what we were looking for, and we still haven't found what we're looking for, to use a U2 song. This is about 1,500 hybridizations in yeast. So what we did was score these hybridizations. Each line is a hybridization. What we did was score them according to their expression profile and if they were enriched in each one of the cluster genes. So this is a self-organizing cluster map, a SOTA map. So once we did that, we found that there was two groups. There was A and AB, which were highly clustered in these, which were highly enriched in these hybridizations, and B and D were highly enriched in these hybridizations. So with two groups completely out of phase with each other. Now, this shows that in every microarray experiment that's on this database, there's 1,500 hybridizations, there's something more general going on than they're looking for. So if you look at the word scores for these, I should actually put this map up. I always keep on forgetting. This is all full of like top hit words like control, time point 0, and so on. And then this group here is always mutant, test condition, and so on. Injection, which leads you to think most of the microarray studies out there are just looking at something incredibly general, not specific. And down below at the bottom here is the actual, this is actually not that good data, but it's the data that was released with the paper. So basically we can see ATP, ADP ratios oscillating between about 1.5 and 5.5 throughout one cycle. So we thought, well, yeah, this is a really good clue to what's happening. And then we produced this simple model where basically we've got ATP increasing, anabolic genes are enhanced, the RSC complex is activated, which causes this enhancement. We've got anabolic gene products and anabolism occurring, which then consume ATP, which lead to growth. But once you produce ATP, you also basically when you deplete the ATP, you increase the catabolic gene production because it wants to produce more. You produce catabolism, then you generate growth stress because or catabolic stress, which is mostly in the form of membrane stress, which I'll talk a little bit about later. And then this inhibits ATP production. So this is a simple dual negative feedback loop which would account for this differential architecture, a differential expression. Now, to get on what I really want to talk about, we started this because we wanted to prove that ISWI and RSC were actually involved in this dual negative feedback thing, which we haven't proved, so I'm not going to talk about that. What we found was when we did the DNA occupancy tests, this is the average of all the genes. I'll show cluster by cluster later, but this gives you an overview of the DNA occupancy. So DNA occupancy experiment, you cross-link, and then you digest, and then you rehybridize it to microarrays, because we never had access to sequencing at the time. The base pair resolution is about 30 on these tiling arrays, so it's not as good as sequencing, but still the message is there. This is the average for all the genes. This is the transcriptional start site here, and then this is the promoter region, and then each one of these lines approximates to a nucleosome. Now, what you see in the system is you've got. I'll show you in the later slide, but you've got various regions where you've got concentrations of nucleosomes, and you have promoter depletion. This big blue hole is a promoter depletion. Now, remember I told you about S phase occurring at this point here. S phase, the switch between S phase starting and S phase beginning, the late S phase and the early S phase. This is occurring right at this drop-off, so keep that in mind. Now, this was all well and good, and really a nice result. When we actually identified the nucleosomes in here, we could see and zoom in on them. The genome body, which are all these guys, all had a very simple, were all being deposited at the same time. So nucleosomes were focusing here in every gene. Now that may be surprising, but every gene's affected. In the promoter side, we've got a triphasic, we've got a triple oscillation in the promoter side. And I'll go a little bit on about to explain how this can happen in the system. So these are the clusters again, A, B, B, C, and D. Now, we couldn't find any difference in the dynamics between any of these. All the clusters were having the same dynamics. So this is the transcription abundance here, the transcript abundance just summed for each cluster. This is the dyad, the nucleosome dyad, or for the gene bodies here. The intensity is different, but the actual phases are exactly the same. There's no difference in phases. The promoter regions all had the same triple point, 3 oscillation cycles per system. So there was no difference between the promoter architecture, no difference in dynamics. This led us to the conclusion that the chromatin is just a substrate for the enzymes, and all chromatin remodelers will act at that particular point based on their cofactors. So that's the conclusion that we drew from this. And the bottom part just shows you the residuals to show the dynamics on each cluster. It shows you the length of the upstream region here as well, this area here, and the nucleosomes, how well they are defined. So this is a well-defined nucleosome pattern here, and this here is a fuzzy one, and this one here is a fuzzy one. Okay, so we then looked more in depth at this using qPCR chip pull downs. This is what you see summarized on the previous graph, but into global anabolic and catabolic clusters. And we can see that the catabolic genes are less expressed, they've got less occupancy, so there's less stuff on the catabolic genes, but that's about the only difference. The phases are all the same, and then we've got this big wave of deposition. Now, to test to see whether it was histone which was actually being deposited at this point, we did H3 pull downs on various blue here is catabolic and red here is anabolic, so various catabolic and anabolic genes, and we can see it was being roughly deposited at the same point. There's a slight phase shift, but the data is from two different experiments, two different periods, but it's accumulating roughly in the same point, and this kind of exists as well. So this bump here and this. So you've got this little bump and then this big bump of nucleosomes being deposited. On the flip side of that, we looked at acetylation, and we've got massive deacetylation here. This is a log scale, and basically at the same point as we've got histone depletion, we've got deacetylation of the system, with these 2 points with these points here relating to points on the sort of here and here, relating to DNA occupancy on the promoter. So when the system's acetylated, you've got the promoter occupied. When it's deacetylated, the promoter is not occupied. Now, what's happening with transcription then is you have the red is the anabolic, remember, and the blue is the catabolic program. The red program switches here to the catabolic program, and the catabolic program is produced. Then you've got a progression and then a drop in transcription rates, not transcription rates, of transcription abundance change. I've got to be careful because transcription rates are something difficult to measure. Of all the genes do this, even the noisy genes. So everything's been shifted off the DNA at that point of the promoter regions, and you have a crash, a reset in global transcription. The Pol2 pull downs confirm this. Here is where we have the Pol2 falling off the system. So the blue is catabolic. There's an increase in the amount of catabolic Pol2, and then during the cycle there's a decrease in the anabolic genes. And then there's a drop down. So the Pol2 is dropping off, and then you've got a transcriptional decrease. And also we've got the AMP, ADP, and ATP here. And we can see that this follows the same trend as the histone H3. It's the inverse trend for the histone H3, the ATP concentration. So ATP is critical for this histone remodeling at these particular points. If you look at what's interesting is this doesn't explain this part of the graph here. So this is happening much later, when S phase is switching and also transcription falls out. This is kind of interesting because this involves a complete switch, but you still need ATP to produce all of this stuff, but not that much apparently, because this is the inferred remodeler activity. The chromatin's remodel by using an active catalytic site where ATP is converted to ADP. However, that ADP on chromatin comes back and binds to the binding site. So it's a very difficult, this is very naive, this approach. This is just a competitor reaction. But the true case is going to be much more severe, but the ADP comes back and binds, and then the remodeler falls off. So that's the kind of inhibition it is. It's a 1 or 0 inhibition. That's if there's too much ADP in the system. But ATP in the system also, if there's too much ATP in the system, it does the same thing, because it doesn't matter what NTP is there. If there's an NTP in between the chromatin remodeler and the cell, it falls off. And that makes sense if you think about how transcription's working and how you don't want to be remodeling an area where transcription's occurring. So if you've got nucleotides there in that area, then that would be a logical thing for it to do, is to fall off if there's any nucleotide in the system, because you don't want to start remodeling an area which is actively being transcribed. So it's thought of as being an inherent safety valve in remodeling. So, but this shows that indeed the transition could be described. It could be just be the activity of this remodeler changing. So this is the ISWI theoretical or inferred value, and this could cause the change in the program. But we're yet to prove this 100%. We're going on to do further experiments. So, but we found the other time when all the things are falling off. So this leads to this more complicated, simple model, where we have the dual negative feedback loop we described before, and then that causes ATP to produce the anabolic genes and inhibit the catabolic genes, which then cause anabolism, which consume ATP, which cause growth. Once the ATP is consumed, catabolic genes are produced. They're remodeled here, and they're acetylated here. At both these time points, they're acetylated. So you've got 2 time points that they're both acetylated. So they're both transcriptionally primed, because acetylation means transcriptional priming. And one's inhibited by the action of the ISW complex, and one's transcribed by the action of the RSC complex. There are other complexes. It's much more complicated than this, but these are the 2 most characterized ones. Once the catabolic genes reach this acetylation state, and once ATP starts to increase again, ATP's kind of start to increase a little bit again, you've got the inhibition of Pol2, the focusing of the nucleosomes. So we've got nucleosomes being focused here. They become very well defined. Once they're very well defined, that creates a big barrier for Pol2, and then this inhibits, this stops transcription. And then it resets back to this catabolic gene system because ATP starts to accumulate again. So here it should be here because ATP starts to accumulate again. So this is the model, the conceptual model we're working on now, and we're actually trying to model it with Reiner. We're trying to get that together. So back to your mitochondria question, if I've got time. This will just take 5 minutes, whoever's mitochondria question it was. This is what the mitochondria are doing during the cycle. These are electron micrographs. This is a flow cytometry measure of mitochondrial mass. During the oxidative phase, the mitochondria, all the membranes, not just the mitochondria are very well defined. The membranes are big and fluffy and full of stuff. So M stands for mitochondria here. You can see this in the mitochondrial mass. The mitochondrial mass measured by MitoTracker Green. It's a live cell assay. The mitochondrial mass is roughly double in all cells, except for this small population here, between the oxidative and reductive phase. Now, when the reductive phase switches, you have a complete change. It looks like a completely different organism. I focused in on one here because they look the same, but you know, they look like the same cell, but they're not, obviously. You can take it into the entire population, and it's common to the entire population. So basically, we can see that the mitochondria and all membrane structures are changing very dynamically. We've done the lipidome on this, of course. It's the first kind of thing you do. Of course, it oscillates. The whole lipid structure's changing, and that was a bit of a no-brainer. When we do the classification on this, and this isn't 100% finished yet, so we're still working on this. These are the oscillating lipids in the system. This is one detector, this is the negative detector. The positive detector's got about the same. In total, I think there's about 200 or 300 lipid types oscillating in the system during the cycle. And we've got a switch between storage triglyceride lipids, which are here in green. This is in the oxidative phase. 2 structural lipids, which are in the purple ones mostly, during the reductive phase. So you've got a switch from burning lipids to membrane lipids occurring every 40 minutes in the system. Now the interesting ones are actually, what I found was the interesting ones is this group up here. This group, the mass match, can only be the cardiolipins, and we've done some two-dimensional on that. And two-dimensional MS is not completely confirmed yet, but it does look like these are cardiolipins. They've got exact mass match. Now, cardiolipins are a very special class of lipids, as they're only mitochondrially bound. And what cardiolipins do is they hold the electron transport chain. This is not a very good diagram, actually. I should have put the other one up. But basically, the glue that holds the electron transport chain in the membrane. So, when either ROS are present, reactive oxygen species, or the membrane starts to be oxidized through eating it, this is what the current theory is about why ATP drops off so suddenly, is the cardiolipins are oxidized, and we've got this in the oxidation state of the cardiolipins here. When it's oxidized, it has roughly this much more mass here. Sorry, this much more mass. So this group in here, the green ones. So when they're oxidized, the mass increases a little bit. And then what we're testing currently is to see whether cytochrome C is released during the reductive phase. And this would be indicative of mitochondrial eating mitochondria. So basically the cell becomes so driven in biosynthesis that it starts to consume all its internal stuff. And it goes to such a state where it just switches to a more biosynthetic format. So this is what we're thinking the mechanism of the oscillation is. This is kind of confirmed with CO2. I'm not really, I just did this data last week, so I'm not really too sure what. We've basically got a switch between, this is a labeling experiment that I described yesterday, but basically we've got rapid switching here and here between consumed, we've got this rapid switch here and then an increase in labeled CO2 and then a decrease in CO2 here. Now this suggests that there's no new CO2 coming in. It's all coming in from the intracell. So there is a big consumption in intracellular stores of of carbohydrate. So right when things start to go wrong, well not wrong, but right where things start to where they start to run out of exogenous energy source, they start using the intracellular energy source here, and then the CO2 label ratio goes down, and then respiration stops, and then you've got glycolysis working. This is what we think. And then there's a lipid consumption here when the membranes start falling to pieces and changing their shape. That's what I'm thinking just now anyway, but I'm open to discussion about that. So there's just a quick outlook. I'm sure I covered most of this. So non-coding I'll not go on about. We've got some experiments, and DNA benders will a little bit about that, but it'd be really interesting to do some experiments on DNA bending. So basically I'd like to thank these people, and mostly Rainer, who did a lot of the informatics with me, Willie and Sabrina, who've done a lot of the metabolomics, and also Kalesh, who helped out a lot during his PhD, and Kaori Igarashi and Alistair Maxwell, who also worked quite a lot on the 13C MFA anyway. So that's pretty much what I want to say, and I'll leave the outlook up because it's supposed to be for discussion, and I've got 5 minutes left, I think.
Would it work to put that in just like batch culture exponential growth? You would expect cycles too?
Yeah, it happens.
You have measured that actually?
Yeah. If you in batch mode during the continuous culture, at the end it starts, when nutrients become low, it starts to talk to each other.
No, okay, but what during exponential? Like, I mean, early exponential, mid exponential?
It's usually glucose limited. It's usually the respiration chains are usually inhibited then, so I wouldn't imagine you'd be able to see it.
Okay, so
in individual cells that are non-synchronized, it has been shown to occur. using fish. So they took groups of RNA that were classified as oxidative and reductive, and then fished them out, and then the fluoride, they were always in opposite phases in individuals. So that's indicative that
Exact oscillations, yes, but not synchronized because
Yeah,
the
yeah.
frequencies are too low and
yeah. Oh, the density. Anyway, it needs to be still under some sort of low growth rate.
okay.
I think low growth rates kind of. Although E. coli is the flip side of it. So E. coli only really shows oscillation at high growth rates and low cell number, which is a bit strange, and I never figured that one out.
There was an Odenan paper and something like that recently showing that an exponential but under high, relatively high glucose,
Yeah.
you would maintain constant growth rate, but you would actually shift away from respiration to fermentation as you grow.
Yeah.
That makes sense, and that's something related also. I mean, it's not oscillations, but
Yeah.
the overall measurements, over population measurements.
It's difficult to. I've done a lot of experiments with E. coli, but nothing that I
Well,
can
that was
really
yeast,
use.
basically, I think.
Yeah, this is always, well, I've grown a lot of stuff, but I like yeast because they smell good and they're easy. E. coli smells bad. So,
You tried mitochondrial-less mutants of yeast?
those wouldn't show this because I've tried them, but it doesn't show anything. It doesn't respire. So, again, that could be a synchrony problem, but I doubt it because if it's correct, then you need this crosstalk between the mitochondria and the cytoplasm to actually occur.
It's not mitochondria free, it's mitochondria DNA free. If it's cell don't have mitochondria, they're dead.
No, yeast can survive.
There are these petite mutants, I
Yeah.
think.
But these have mitochondria, but they don't have mitochondria DNA. That's a very special, very important distinction.
No, they generally don't have the mitochondrial structure as well. They've got peroxisomes.
No, they have this membrane. They definitely, because they need the iron sulfur complex to do all these.
They're very sick, but they do grow. They need molecular oxygen, but they don't have mitochondria as far as I've seen. And we've looked at them under the electron micrograph. Maybe the mitochondria are so weak that we can't see them. But
That's what I heard is that you cannot. I mean, the yeast folklore is that you cannot never get rid of mitochondria because of all this. Also, like the amino acid synthesis also happens in mitochondria, and also because of iron sulfate complex. So they lose DNA, but not the mitochondria. But I'm not sure. How these
well, they might be so weak that it's difficult to visualize them under electron microscopy. So, but I've never seen any mitochondrial structure in the row zero stuff that I've been looking at. But no, to answer your original question,
So in yeast you need the mitochondria to have these oscillations, but you have the oscillations in E. coli and they don't have mitochondria, so is this a different mechanism?
but they've got an electron transport chain, so I think probably you need an electron transport chain. So yeast without mitochondria are basically yeast that don't use electron transport. So I think you need the electron transport chain in order for it to synchronize. But it means I haven't proven for sure that it's the same oscillation in E. coli than in yeast. That would take a lot more effort on my behalf to do this. It would be nice if it was, but it's got more resources than I've currently.
Optimality. So what would be the evolutionary forces driving something like that?
Well, that's an interesting question. We did originally think it was DNA division being out of phase with reactive oxygen species generation, but that's not true. That's just not true because DNA synthesis can occur anywhere in that cycle, so that's simply not true. That was a nice theory, though. What we're looking at now is that certain groups of reactions perform much more optimally because the pH oscillates here as well. So under certain intracellular conditions, you can really maximize one set of reactions way beyond the average for the average cell pH and redox and ATP availability. And then on the flip side of that, you've got other reactions which are And if you're changing the cellular environment in an oscillation, you can probably get an optimal solution for every enzyme there is.
Okay, so basically going towards temporal compartmentalization.
Yeah, temporal optimization. So we're going to do this now, and we're going to do the best we can. And then later, and then that has been, I think that's a good idea to increase yield as well. I think that would actually increase overall, because some reactions operate at 10 times more efficient when they've got double the amount of ATP there, for example. You know, it depends on the thermodynamics of the reaction.
Yeah, issue would be, I mean, you could imagine that that at the same time you might have selected forces that would drive you into a direction where you can do everything at the same time, because that I mean keeps you out of the mess basically. You don't have to do that all the time.
Yeah, well,
I think there are arguments that speak for temporal compartmentalization, including like also intermediates.
Yeah, incompatible.
Might be constrained and might interfere with other stuff and stuff like that. So, but it would be fun to show that in a very explicit way.
Yeah. We're thinking about that currently, for sure. We've got some quite advanced ideas about that, and we're trying to put down some framework for both computationally and and experimentally testing it for sure. But the concept that it does enhance reactions depending on their optimal conditions, rather than keeping everything the same, is quite attractive.
Yeah, why not spatially separate?
Because that's just not enough sometimes. You'd have so many compartments in the cell that what you would do would be land up making biomass just to keep these compartments going when there's already, I'm sure it's a trade-off between making compartments and temporarily separating everything. I think that's what I would say about that. Because it does take a lot to make a mitochondria of resources. So why not just change its shape for a bit? That's what I think anyway. Probably wrong. Been wrong before. It will happen again.
That's the only fate a good theory can have, is being proven to be wrong.
Yeah. Well, I really did like the reactive oxygen species in DNA theory. I really did like that, but it's not true. So, oh well. Okay.
Thanks.
Okay. Thanks very much.